I'm going to talk about NIH from these two angles of approach, or two contexts. Really, the discovery of new knowledge and the ways that those discoveries move to enhance health. Um, those, I think, the key driving forces. The NIH has been called the crown jewel of federal spending. Why would that be? That's a big claim. It makes discoveries that turn into health, that literally turn into health. Seek fundamental knowledge about living systems and advances that knowledge that was been discovered toward enhanced health, longer life, and reduced illness is sort of a summary of the mission statement of NIH. It gets a lot of money, invests billions of dollars to support biomedical research. It is a lot of money, but I like this quote from Mary Lasker. If you think research is expensive, try disease. And the important point there is that $47 billion is about 1% of the health spend in this country. So any industry that only put 1% of its funds back into research would not last very long. So it's in that sense, it's a bargain. About 80% of the NIH's appropriation goes out of Bethesda, out of DC, to 2,500, more than 2,500 universities, medical schools, research institutes, and to hundreds of service, supply, and instrumentation companies employing about 500,000 people in your districts and states and contributing over $90 billion to your local economy. So even as just an economic driver, the NIH is significant where you live and work and in the regions that you represent. So how does it do that? How does it make discoveries, turn discoveries into health? And it's complicated, and so I'll try to take you on a little tour through that. First of all, how is the NIH organized? It's the National Institutes of Health. The NIH is a loose federation of 27 separate institutes and centers. It's one of the agencies of the Public Health Service, which, of course, as you know, is part of the U.S. Health and Human Services Department. Wonderful federal bureaucracy that we live in. 22 of those institutes and centers are funders. They're funding agencies that give grants to investigators, to institutions that are carrying out the work that is the topic of those agencies that you see. And as you scan that list, there's a bunch of them that will be familiar to you. So the National Institutes of Health is this federation of funding agencies and other centers. One of the centers that I'll talk about is the one in green, the Center for Scientific Review. And I'll come to the work of that very important center as we move into the talk. So each institute receives its own separate appropriation, and then the sum total of that, plus the money that goes out to the centers and to the office of the director, is the total of that $47 billion that we talked about. How do they decide how to spend their money? So prioritizing the funding in each of these funding institutes is complicated and difficult because so little is understood about how biology works. So they don't know enough, we don't know enough, to be able to say, oh yeah, we know exactly where to spend our money because here's the question we need to answer. So instead, there's kind of a general separation of types of research that the NIH spends. And this pyramid is really important because you see that the base of it, the solid base on which everything else stands, is basic research. Fundamental research that's just designed to understand something about some biological process. So over 60% of the NIH's funds go into basic research. About another 25% is directed toward so-called translational research, the kinds of research that bridges the foundational knowledge that's understood in the basic science side and begins to understand its relevance to something on the clinical side. And about 15% then is straight clinical research. So Vannevar Bush is the guy. He was actually the first U.S. science advisor. He was President Roosevelt's science advisor. And he wrote a very famous report called Science: The Endless Frontier. And in that, he said that basic research provides scientific capital. And we'll talk a little bit about what he meant by that. Now, the catch about basic research is that only the government can really support a substantial basic research endeavor because it is untargeted. Right? It doesn't start out with a goal to try to understand cancer or arthritis. Right? It starts out just saying that a scientist has a curiosity about some funny biological process that she's observed and wants to understand it better, so that individual designs a grant proposal that goes to the NIH, says, here's what I want to try to study. But it's not targeted to a specific disease or syndrome that's health. So that means that the government has to support basic research because industry can't. Industry has to focus, right? And so it can't just be wandering around and saying, oh, what do you think is interesting, what is amusing in front of you that you observe today? So government has to do that. And Bush made that point in his talk. And that's sort of the founding principle for government support of science and technology in this country that was actually set in 1945 and pretty much still exists except for what has happened in the last 2 years, thanks to the efforts of NIH and others to stand up this new agency, ARPA-H, that I'll talk about in a bit. So it's really discovering fundamental knowledge about biological processes. And there's so much unknown out there that we don't even know what we don't know. It's not like we can make a list of the things that we need to understand and then we'll be there. We don't even know what is out there that needs to still be uncovered. So why is that? Why is it that difficult to even map out what it is the kinds of questions that we should be asking? And the answer to that is that biology is complicated, and it's complicated for a good reason. So here's 3 pathways, 3 processes, right? One of them unidirectional, the other 2 are bidirectional, things that everyone's familiar with, right? Things that actually happen, biological processes that actually happen in bodies or in a developing organism. So these processes are all encoded in our DNA. DNA doesn't change, so it's hardwired, right? But the way that the processes play out is not hardwired. That's interesting. I mean, the DNA code is there, and it's saying how things should work. But the way that each of these processes plays out, difference in different people at different times, right? Despite the fact that the DNA is just there, the code is just there. So that's interesting. So that means that these can't just be simple switches on or off, and they can't even be, these arrows can't even be hiding the fact that there's multiple steps along the way. Because something is making these processes play out in ways that are affected by people's experience and individual physiology and so forth. So the good reason that biology is complicated is that if everything was hardwired, right, then we wouldn't be able to respond to differences in physiology, in environment, in your experience. And so the ways that these processes work need to be adapted, changed, modified according to your own physiology and experience and environment. So, okay. So instead of this simple diagram, what we actually have is this. Basically a plate of spaghetti, a bunch of really complicated pathways. So let me take you through this. So down at the bottom, not actually shown, are the genes. So this is a cell. Here's the nucleus where the DNA is, the genes are, right? And then out in the cytoplasm and then outside the cell. So the genes are coding for those biological processes we talked about. Let's see, levels of blood sugar, whether the cells are going to divide or not. And all those things, all those rules are then encoded in the DNA. But the way that the genes are expressed, ways that they're turned on, turned off, turned up, turned down, right, are dependent on this plate of spaghetti. So what's going on up here? So out in the cytoplasm and in the cell membrane, right, are a bunch of proteins that are called signal transduction factors. What are the signals? The signals are molecules that are swimming around in the blood, or actually inside the cells, small molecules that interact with some of these so-called signal transduction proteins. So the most obvious ones are these sensors, things that stick through the cell membrane and actually are communicating with signals coming in from the outside, right? But there's sensors inside the cell as well. And those sensors, when the signal comes in and hits the receptors, the sensor, and triggers its activity, it changes the characteristics of the proteins that are associated with it, right? And those then say, oh, I've got this signal that says my person is thirsty, tired, maturing, right? And the characteristics of those proteins change. And they go in and they interact with other proteins that are receiving other signals. So it's the combination, the particular combination of signals that are triggered that lead to different kinds of interactions inside the cytoplasm of the cell, right? And that combination of interactions at any one time then leads to a change in activity of some proteins inside the nucleus that bind to your friendly gene and determine whether it's going to turn on, turn off, turn up, turn down. So those hardwired genes are expressed differently depending on the combination of signals that are coming in. All right. So now you can see things get a little complicated. This is just a tiny little fraction of all the stuff, the plate of spaghetti that really exists in the cytoplasm. So the result of this is that biology is really complicated. And the result of that is that disease is really complicated. Mutations that affect any of those sensors or underlying protein networks then alter the eventual expression of genes, which then alters the risk of getting a disease or the progression of disease if you get it. So the genes for any of those proteins on this diagram, or then going out to the rest of the cell, could actually be seen as identified as disease genes because they're changing the potential for getting or how serious the disease would actually be. So things get really complicated really fast. And it's really the reason why we need to do basic fundamental research just to understand processes and that we don't yet know what we don't know. So corollary of this complexity is that fundamental biological processes are often solved in simpler organisms. Simple organisms contain fewer layers of these sensors, right? So as organisms get more complicated, the way they get more complicated is all of these networks get more complex. Simple organisms allow for more definitive experiments. You can imagine there's lots of experiments you can't do on people. And complex organisms, take you for example, evolved from simple organisms. So what we learn in simple organisms usually teach us something relevant to something about us. There's plenty of places to sit. You guys come on in. Or if you look at me. Okay. So this actually works. This notion of working with simple experimental organisms to learn something relevant about these complicated processes actually works. Here's a list of examples. Some sources of breakthroughs in our understanding of some of these processes that scientists would like to understand came in simple organisms. Understanding gene expression, gene regulation, something that I worked on my whole career, started in bacteria and their viruses. Not viruses that affect you, viruses that infect the bacteria. Cell division in yeast and the clawed toad, cancer genes in chicken viruses, development in the fruit fly, sea urchin, and fish, aging and lifespan in soil worm and my favorite, pond scum, learning and memory in the sea snail, and neuron target connections in the chicken. All right, so that's really good. It's really instructive that we can actually learn things relevant to us because of evolution by studying simpler organisms. Now you may look at this list and think, but wait, won't taxpayers consider studying sea snails and pond scum to be a waste of money? And here's something really interesting that I wouldn't have expected. That it's an organization that I'm associated with called Research America, does polling of the public about their attitudes and knowledge and appreciation for or not of science. And one of their questions that they ask, and this is just verbatim what the question that's asked, is this: agree or disagree, even if it brings no immediate benefits, basic scientific research that increases the frontiers of knowledge is necessary and should be supported by the federal government. And 80% of the responders in this survey either strongly agree or somewhat agree. So that's heartening and something that I think that you should kind of take home with you and be aware of as you or your member go out and talk to their constituents. And not be nervous about supporting fundamental research done by the NIH. Nevertheless, when you talk about untargeted discovery, it sort of sounds sort of random. I mean, people are just out there bumping into things, or what's going on? So it bears asking the question of how is it that these things are decided? Who chooses what to study and how to study it? Right? So the predominant NIH funding tool is something called the Investigator-Initiated Award. And that really does say, just sit down at the terminal and type out something that you think is interesting and send it in as a proposal to get funding from the NIH. So with this mechanism, NIH literally invites scientists to propose what to study, how to study it, right? Driven solely by their curiosity, imagination, creativity, vision, and talent. So hammer out this thing, send it in, an idea that they think is interesting. And then the NIH takes those applications and gives them to other scientists, other working scientists, so not infrequently competitors of that person, and says, this person sent in this grant application, what do you think of it? And based on the answers, NIH decides to send money or not. You think, that's a wacky system. I mean, the ways to think about conflicts are pretty easy, right? So who makes the priority decisions about what to do? The scientists themselves. And how those decisions are made is this process called NIH peer review. All right. You've seen that phrase all over the common literature these days because of what happened during the pandemic with scientific papers that were submitted for publication, whether they had been subject to peer review or not. Well, the NIH grant applications are all subject to peer review. What does that mean? What is NIH peer review? How does it work? So peer review is what we just saw, evaluation by scientists of the scientific merit of the proposed research of other scientists. And over 80% of the NIH budget, that big budget, is allocated in this way. So that green center that I highlighted, the Center for Scientific Review, CSR, which is independent of any of those 22 funding institutes, right, manages all of those proposals that come in, 80,000 of them a year, using 20,000 reviewers, 20,000 working scientists who volunteer to come to Washington and sit in a room and make judgments about proposals of others. The reviews are executed by these committees called study sections, committees of experts in a defined area of study that are made up of working scientists who volunteer. They're virtually all other NIH grant holders. So there's 5 criteria that every reviewer has to comment on in writing his or her review. These are close to my heart because I proposed these 5 in 1995, 28 years ago, and took 2 years to get them approved. And so they've been installed in part of everybody's grant applications since 1997. Before that, there were no criteria, and the reviewers just said, I don't like the magnesium concentration that this person is using, so no money. All right, so what are the 5? Will it make an impact? Will it make a difference in the way that the field is thinking about this process? Will it advance things in some way? What about the approach, the experimental design? Are they good experiments? Will they work? And will they be telling? Will they inform the process? Is there something new about the way the person is thinking about the problem or approaching it? Is the investigator qualified to do the work, or does he or she demonstrate that the things that he or she doesn't know, right, are going to be covered well by a collaborator that they've recruited and they can name in the grant proposal? Is the environment, the institution in which the person's working, set up in ways that will facilitate achieving the goals of the grant application, and so forth? Okay. So every reviewer has to come in on each of those 5, and then having done that, then make then assign an overall score, ranked integer scoring 1 to 9, top is 1, bottom is 9. And the average of those scores is, for each of the members of the study section, that big group of people around the table, right, is then computed, multiplied by 10, and then that is the score that is assigned to that priority score that's assigned to that grant application. You can see the descriptors of the scores 1 through 9. And I'll tell you that things are so competitive that the grants that actually get paid are all scored between 1 and 2. It is really tough business here. You know, having an excellent proposal, 3, pretty much out of luck. So that's how competitive things are. I'll point out later that really only a small fraction of the grant applications actually get funded. So that's NIH peer review. It's imperfect, and I'll point out if it's imperfections in a second in this slide, but very impactful. It really works, all right? It operates on this massive scale, 80,000 applications across all of biological, biomedical science, right? It somehow manages those conflicts that are easy to think of. people that you're competing with? Wouldn't it be nice for them not to get their grant? Right, and so forth. Yes, it does tilt a little bit toward the conservative, right? Because the working scientists in the room are pretty much working along doing the ways that their colleagues and friends are working. And so they're going to like that kind of application. So it tilts a little bit to the conservative. It generally aligns with prevailing paradigms because, again, the people in the room have been involved in making the prevailing paradigm. So they're going to like a grant application that kind of agrees with what they've been working on. And study sections lack sufficient diversity. There was a great paper published 2 years ago in Cell called Fund Black Scientists. And it was that was specific a topic line because a study done by Donna Ginther in 2011 that showed that black scientists are underfunded in the NIH system after controlling for every variable that the social scientists could take out. And not much progress has been made on that since 2011. So there's work to be done. And really, at the end of the day, the only way to really solve that problem, aside from raising people's consciousness, is to increase the diversity on the study sections themselves. All right, so yes, there's good stuff, and there's stuff that's problematic, but it works. Remarkable things happen. Here's a great and simple example. Here's 2 proposals that came to the NIH. And both of them you might think sound sort of at least esoteric, if not wacky. One of them said, this person wanted to sequence bacteria that somehow managed to survive and thrive, in fact, in these acidic mine drainages, right? Superfund cleanup sites. And so this person said, gee, that's interesting. How do those bacteria do that? So will the NIH give me some money to sequence their genomes? And the study section said, yeah, okay, go for it. A second proposal, completely unrelated, said, I want to study a bacterial immune system. And you think, all right, well, that's cool. I mean, how will it learn about our immune systems? And the person would have to say, it won't. It's completely unrelated. And the NIH said, okay, go for it. And when those people completed their work, they didn't complete them at the same time, but let's imagine that they completed them at the same time. When people completed their work, people stepped back and looked at the results and said, oh, we can really do something amazing here. The amazing thing that they did was to generate the CRISPR system that you're aware of that's used for editing genomes. All right? So 2 basic science discoveries, unrelated, produced then a powerful new basic science tool, capacity to edit genomes to do experiments, that had enormous clinical potential and, by the way, enormous ethical implications. Right? But this process of these unrelated grant applications that come together to lead to something this significant is there, and it says that the process really works. Is it the best system? Well, what are the alternatives? Well, you could have a central committee, this actually happens in some countries, that sits down, a big, old white guys that sit down and decide what work is going to get done in the country. You'd have a hierarchical system of the Herr Professor that then rules a big battery of lower professors and trainees that says, here's what we're going to work on. Or you could just take the money that you have available and divide it up evenly among all the scientists in the country. None of those works, we've seen by experience. So in fact, by any measure, NIH peer review has produced the world's preeminent biomedical knowledge discovery engine. By any measure. So here's one measure, Nobel Prizes. There have been 230 Nobel Prizes ever awarded in the world in physiology, medicine, or chemistry. Of those 230 Nobel Prizes, 99 have gone to NIH-funded scientists, to 163 NIH-funded scientists. It's not too shabby in the dominance category. Okay, so that's good. It says that this system that NIH has crafted is really good at knowledge discovery. But is NIH-discovered knowledge really addressing disease? So here's one version of yes that happened to all of us during the pandemic. And that is that during the pandemic crisis, it took only months to create powerful diagnostics and tracing methods, therapeutics, and vaccines. So it was actually stunning, right? How did that happen? It happened because of decades, many decades, of untargeted basic research that had accumulated information that could be sewn together in the same way that those two unrelated projects we just talked about were. So that says that this discovered knowledge idea, this untargeted research idea, really does work in crunch times like this. So that's one version of yes to that answer. Here's another one. So untargeted NIH research, it turns out, has been demonstrably essential for new drug approvals. This is a paper published in 2018 that looked at the 210 drugs that had been FDA approved over a 7-year period from 2010 to 2016. Right? And it turned out that NIH-supported research was associated with all 210 of those approved drugs. And 90% of that NIH funding was for untargeted basic research. Right? Pretty impressive. However, industry can't do it alone. Government support, coordination, and management. Support, coordination, and management are essential in order to accelerate the application of new knowledge. So what's hidden in that all 210 drugs, what's hidden in the progress in COVID-19 treatments is the fact that things are slow because industry can't pick up these things and pay for them in an efficient way. Of the 9,000 human diseases, you know that only 500 have approved treatments. And then there's this study. The 24 most impactful drugs on the market all stemmed from untargeted basic research, but the median time from discovery to FDA approval, the median time was 32 years. Not so good. So this is where ARPA-H comes in, what Anna talked about. So this is a new federal biomedical agency. It was launched by Congress last year, thanks to Anna's work. NIH and ARPA-H are separately appropriated. They operate very, very differently. So the goal of ARPA-H is to accelerate application that comes from NIH discovered knowledge. But ARPA-H, the goals don't overlap. ARPA-H's goal is not to discover knowledge itself. So NIH's knowledge endpoint is ARPA-H's starting point for developing platform technologies, devices, and therapeutics. ARPA-H promise is only a year old, but ARPA-H promises to take risky strategies and approaches in order to move things quickly and efficiently toward applications of new knowledge that extend and improve life. And this is a little a slide that I stole from the NIH, the ARPA-H director, Renee Regson, and just shows some kind of big aspirations of what ARPA-H can try to take on. So our NIH and ARPA-H will operate in tandem to discover and then apply new knowledge. So it comes back to NIH. NIH discovered knowledge is the essential start point that leads to conceptual and technical advances, understanding of biological processes, insights into mechanisms of disease, and at the end of the day, enhanced health. So this ongoing commitment to excellence offers the continued promise for extraordinary scientific discovery, but it's expensive. Paying for it, paying for great science, is difficult and problematic because it's expensive. So we really need continuous increases in the NIH budget in order to carry this work forward. We can talk about what you may ask later about why the continuous increases, but we can talk about that. Only 1 in 7 meritorious grant applications is funded. Only 1 in 7 of those grants that are scored highly in that scoring system actually gets money because of the degree of competition. This means that great opportunities advances are almost certainly lost. Nevertheless, $47 billion is a lot of money. So it's fair to ask, it's fair for you and your member and your member's constituents to ask, is there really a return on investment for this? So this expenditure of my tax money from your constituents. And that's where a test that I generated called my Starbucks test comes into play. This is a test you can give to your members, to yourself. You're going to give it to you in a minute, and constituents. So here it is. Over the past 40 years, cardiovascular disease mortality has declined by 60%. That calculates out to 1 million fewer deaths per year. So, what was the NIH investment per American resident per year in $5 Starbucks beverages, which are becoming rarer, I have to say, that saved 1 million lives a year? How many $5 Starbucks do you think per resident per year? Any guesses? 1. Would you give up 1 Starbucks per year to save 1 million lives? Just think what could be done if we gave up 2. So that's it. NIH is really the crown jewel in federal spending, in quality, efficiency, and impact. Really thanks to the volunteer efforts of scientists to identify the best research proposals and to funding from Congress. So tell your members to join the Congressional Biomedical Research Caucus. There's the link. And support increased funding for NIH research to institutions, trainees in your district and state. Questions? Comments?
Is there any specific parameters that the NIH is hoping for in the annual funding increases?
Is there any specific?
Like you're asking for a support for increases. What's an example?
Yeah. So actually, the last 8 years, Congress has provided bipartisan support for NIH. This is, as you well understand, goes through multiple administrations, multiple different parties in power in Congress. And they have been increasing the NIH budget on the order of $2 billion a year. This year looks different. You know, the reason this is important, the reason this is important is that when NIH gives a grant, I should have said this, the grants are for 4 or 5 years, because basic research takes a long time. And so then NIH says, all right, here's 4 or 5 years of money. And you recognize, of course, that what you guys do is to create federal budgets on a year-over-year basis. All right? So that means that every time NIH gives a grant, they're making a bet that Congress is going to stand behind them and provide that money every year, right? This year looks different, as you all appreciate, right? Right. The House mark was $2.8 billion below this year's instead of $2 billion above. Senate mark is about a $230 million increase, much smaller than usual, but not a decrease. So we'll see what happens when the budget gets negotiated in conference, and we don't know that yet. But we haven't seen a threat like this in a very long time. And it would be a very, very serious thing if it were to happen.
I think one thing to add to that is
Lynn.
with the fear of a shutdown, science doesn't mark where you can stop and pick it up, 3 weeks later. It's a continuous thing that has to be tended to and worked on a regular basis. It's hard to do stop and start, so shutdowns are particularly damaging to science and the scientific enterprise.
And you all know that the risk of a shutdown is very serious.
Can you describe further on what would happen on an institutional level for a lot of the people that rely, like in universities, on grants that come from the NIH?
Yeah. So you hear the question: what would be the impact on people in universities and similar institutions that rely on NIH funding for their work? So there wouldn't be an immediate cessation of the funding because it would be out to wherever the grant birthday is for that individual. But in fact, the studies would really be at risk. And if the government were really to shut down, then that would really be problematic because the money would stop going out the door. So the impacts would be dire.
So for the biomedical research caucus, is it more government oriented or is it more science focused? So like, if let's say I were to join, right? Could you take on a role? Like, what are the kinds of roles? Like, is it a lot more for policies, or for people who are interested in doing research, or is it people who are just advocating for like the research and like speaking at the conferences?
Yeah, so it's a really good question. It's very broad based, and what the caucus does is to help define for basically programs these meetings is one of its main activities. And so members of Congress can make requests of the topic they would like to see covered, right? Or the caucus can actually make proposals to Congress, or actually just invite somebody to do this. So this talk, for example, we tend to present in each new Congress year, right? Because there's new members that aren't familiar, don't know what the NIH stands for. That can actually learn something about it. But it's not all government, so your question is very good. It's not all government focused. And it can actually we could actually have people visiting and working in areas to work on rare diseases, for example, right? And you know, this is a major problem area because rare diseases, by being rare, affect a relatively small population of people. But in total, the number of Americans with rare diseases of one kind or another is like, it's many, many hundreds, a couple hundred million worldwide. And so there's topics like that really need to be stood up and discussed and considered. You know, how do we manage that? Well, we know that industry can't go after a very rare disease because there's no profit there. Right? Industry is not going to go after diseases that afflict parts of the world, countries that have no resources because there's no profit there. So how do we manage this and begin to build some equity into the system? So topics like that are things that the caucus can also take on. it would be great to have your voice involved.
Hi, Hannah Fry with the MIT Washington office, and thank you so much for your presentation. And I can say, as a past scientist who got those 1s, 2s, and 3s and still didn't get funding,
Yeah.
can attest to like how competitive this system is. But would it love to hear a little bit more if you could elaborate on ARPA-H and some of the value that it will really drive towards taking those like NIH basic research and really putting it into health innovation in the hands of patients, hopefully more quickly than the current process.
So the way that it's a great question, Hannah. The way that ARPA-H, its operating model, is very different from NIH's. So an NIH grantee, you, are the controller of the way the question is asked, the way that you approach it, and so forth. And it's for fundamental knowledge discovery. In ARPA-H, ARPA-H invites people to come forward to the director and say, I want to be a program manager. I have a big idea about something that I could accomplish in 3 or 4 years, right? That will actually have an impact on people in that 3 or 4 year period. It will require my pulling together a team of people, some of whom know what I know, but more importantly, people with very different expertise: engineers, material scientists, computer scientists, chemists, physicists, so forth. And if I do, and I know some of those people and don't know others, I'm going to need your help to identify them. But I'm going to put together this team. I'm going to run the team. The project manager that gets recruited in that way then is given a big pile of money, $50 to $150 million, by the ARPA-H director, and says, all right, you convinced me. You can do this. Here's $150 million. You have 4 years. Go get the job done. And the ARPA-H director is on top of those program managers all the time to ask how things are going. The program managers are on top of their team contractees all the time, really meaning phone calls every couple of weeks, right? And if things are going well, if things have gone well, but they need more resources, the program manager can give them more money, right? If things are going badly, the program manager can stop the money that day, right? So it's really brutal in that sense, but it really keeps things moving and on track. Whole programs can be terminated, and new programs, then resources are available to start new programs and so forth. So it's a very highly managed system made up of teams with multiple disciplinary expertise that can then together take on a problem and have a product at the end. The products are not things that go onto store shelves right away. They're prototypes. But they demonstrate to industry this is now something that can be done. So it's a big de-risking exercise for industry to doing the stuff that industry can't afford to do because of the risk involved, getting all that stuff out of the way, and then coming up with a prototype that industry can say, all right, I can make this and get it out. Right? So that's a big difference. Okay, it's 1 o'clock, you guys.